Welcome back. We continue our discussion on atomic force microscope. This should be probably the last lecture on AFM. There, as I told, AFM is a vast topic and it's still developing and evolving in various settings, but this is just a preliminary course to give you an idea what is an atomic force microscope. So I will limit my discussion essentially to the basic modes. Let's see if something continues. We'll in the next lecture we'll just wrap it up. So this particular slide should now make sense to all of you. It's essentially some sort of a very improvised schematic of a constant force contact mode imaging, and this moves from point 1 to point 2 essentially during the rastering, and we also now understand it's the piezoelectric scanner that essentially takes care of this movement. So this also you should be able to sort of understand now. This is essentially tells you how that rastering is done. Depending on which direction you are rastering, you can choose your fast axis and slow axis, then you essentially get a data. Some will be plus and minus, and sorry, this will be 0, the first point was 0 because that is the datum. Anyway, before I move on, there are certain issues with the contact mode imaging, and the most significant issue is something I mentioned briefly. During the rastering, you are actually dragging the tip over the surface. So that is detrimental for both the tip and the surface, because of the friction, the tip sort of becomes blunt pretty soon, as well as if the surface is soft, particularly for polymeric material, contact mode imaging often leads to scratches on the sample surface, damaging it, so you cannot use it. So there has been development in this so-called non-contact imaging modes, particularly the intermittent contact mode or the tapping mode. But before I move on, I would just like to draw your attention to one unresolved topic which we discussed during during our discussion on soft lithography, and I have just borrowed this slide from our lectures on soft lithography. So now all these images, in fact, you understand these are all atomic force microscope images, and you also understand whether it is green or brown doesn't make any sense, because all color in an AFM image is artificial, because it's only the color base you choose during when you convert your data into the grayscale that in fact matters. In fact, both the images, to be honest, are incomplete because none of them comes with the vertical scale bar, and therefore when I was rendering these images, I have missed out vital information about the height of the structures. Anyway, coming back to context, so now topographic pattern, we understand that they can be imaged very conveniently using an AFM, and you can get all the relevant information like their profile as well as their feature height, which is extremely important in the context of nano patterning or any surface patterning technique. But a question still remains unresolved. We talked about this micro contact printed surfaces, right, chemically patterned surfaces, and we argued that these surfaces have virtually no topography, but it has domains of different wettability like hydrophobic, hydrophilic, or different wettability contrast regions. So how does one image that, or is it even possible to image it? Because as the tip moves from point 1 to point 2, it is virtually flat. However, what is different between, let's say, point 1 and point 2 in this type of a surface is these areas are covered with something different, some species that offers a different surface energy, and this different surface energy, thankfully, is often associated with different frictional properties also. To put it in very simple terms, and that in fact one can very cleverly use in contact mode imaging to generate an image of a micro contact printed surface. So let us see what we are essentially talking about. We are essentially talking about a surface which is physically homogeneous, but chemically there are different phases. So it's a smooth surface, but there is some area which which comprises of different material. So here you see the material is different. So now what happens? What happens is, though we have been talking, or even this image shows that the tip sort of is moving in this direction just like that, reality is you are in fact dragging your tip during this rastering action over the surface. And please do not forget that the tip is in contact with the surface, and it is the scanner, because of the contraction or expansion of the piezo, that wants to move. So, it definitely moves, and as it moves the tip will also move, but since the tip is adhering with the surface, depending on the frictional properties or adhesive properties of the surface, there is some sort of an adhesion which translates to some static friction, which the tip has to overcome in order to move from point 1 to point 2. Now, if the material of the surface is uniform, then this, so what is this static friction going to do? The static friction, what it does is the tip does not move like this. So, if the tip is moving in this direction, it undergoes some sort of a torsion, some sort of a bending, right, because of the fact that this end is moving while this end is sort of still stuck up on the substrate surface, right. So, if what I am trying to emphasize, if the surface is homogeneous, irrespective of the topography, if the surface is physically homogeneous, irrespective of the topography, this lateral distortion or movement of the tip is uniform across the sample. But now you can imagine that when your surface has different domains, different phases, this frictional resistance or the local adhesion, extent of local adhesion is going to be different, and therefore, the lateral displacement, let us say on these blue patches and these green patches, are going to be different. By now, you have probably understood that scanning probe microscopes are all about finding out some sort of an error. So, you see here you can find out or pick up an additional error, an additional difference in terms of the extent or the magnitude of the lateral displacement, and that is exactly what is shown here. For example, on this light blue patch, the image is not fully correct. One should have shown this to be also partially distorted, but that is something missed out. So, you can consider that these areas have very low adhesion, which might be possible if you have a very low surface energy coating, but when it reaches this particular area, you see the adhesion is more, and therefore, the resistance in the form of static friction is higher, which in turn leads to more lateral displacement of the tip. So, you can essentially generate a sort of an additional contrast, not only in the deflection of the tip from point 1 to point 2, right, because of the topography of the surface, but also depending on the chemical composition of the surface from point 1 to point 2, there can be additional lateral displacement or bending of the tip, and this you can pick up as a contrast. And this you typically, if you look into an AFM scan, you will find that there is a window which gives you the frictional map, the frictional image, and this is nothing but the lateral displacement or the lateral force. So, what you are in fact seeing is a very interesting image. This is in fact the image of a micro contact printed surface. So, you see the topography is nearly flat. It looks rough, but look at the vertical scale, it is plus minus 0.05 nanometer. So, all this roughness is confined to 1 angstrom, which means that the surface is extraordinarily smooth. But when you look at the corresponding frictional image or the frictional image, you find that there are different domains, and then these different domains correspond to different wettability areas because of micro contact printing. So, this is what is known as one of the secondary imaging modes, but it is omnipresent along with whenever you are doing contact mode imaging, this is omnipresent. This is what is known as the lateral force microscopy. So, we are now about to finish contact mode imaging. So, advantages is you can do reasonably high speed scan, you can get the highest resolution and can image fairly rough samples, but there are disadvantages since the tip is in contact with the surface, and typically AFM does not require any sample preparation or imaging is done in ambient condition. I mean, you do not require any vacuum chamber or something like that, but since a sharp tip is always in contact with a surface, there is always a tendency, if your lab environment is humid, that a capillary bridge might form, and that sort of reduces the resolution. Something that I have already mentioned, that contact mode always has a tendency or possibility of damaging the surface as well as the tip on rough or very hard samples. So, The development essentially was the non-contact mode imaging, which I essentially pointed out that you do not go all the way here. You would like to keep your tip few nanometers away from the surface and do something. So, if you are few nanometers away, for example, if you are here, the tip will have a configuration like this, and you can raster it and try to track if there is any change, but that is seldom done. What is done in non-contact mode imaging is one takes advantage of the fact that the cantilever nearly behaves like a spring. So, there is in fact an additional drive, so which oscillates the cantilever. Typically, the oscillation is done at the resonant frequency of the cantilever spring. Why? Because at the resonant frequency, the oscillation amplitude is going to be maximum for a given amount of power that you are giving. So, contact mode you already know, where there is no oscillation, you simply bring in the cantilever in contact with the sample surface. Typically, in non-contact mode, in classical non-contact mode, what you do, you bring the cantilever very close to the surface and then start oscillating it at its resonant frequency. You can choose the amplitude, but you choose the amplitude in such a way that the cantilever does not touch the surface, right. So, what happens is, here is your surface, you bring in your cantilever here. So, you somehow stop your approach here, how you stop it, we will discuss, and then you start oscillating it. Frequency, let me remind, is the resonant frequency of the cantilever. So, that is why it is sort of a material property, but the amplitude you can choose. In typical non-contact mode, let me repeat, you choose an amplitude where the oscillating tip does not touch your sample surface. So, now what happens if you now do rastering under this condition? So, what happens is that as you go away, the net force sort of changes, and as the net force changes, because the force that is acting between the tip and the cantilever is lower here, and you look at the resonant frequency of the cantilever. So, which is the effective, because what is important is the effective force constant in fact changes. The effective force constant changes because the delta F is the change in the external force, and which sort of changes, and you would like to keep the amplitude constant. So, what happens is, as the effective frequency changes, the amplitude changes, and now what you need to do, your piezo gets activated. So, it now again becomes slender and it pushes this a bit downward, so that your amplitude matches with the amplitude at approach. So, this is in a nutshell the typical non-contact mode imaging, but getting a good resolution is very difficult because the tip does not touch. Of course, it is scientifically very rich because you are in the attractive regime, you never touch and you rely entirely on your forces. Unlike here, where you are in fact in the deep repulsive regime, you are not really relying on an attractive interaction between the tip and the sample surface. So, I am deliberately sort of rushing through non-contact mode imaging because that is not an industry standard anymore. What is the industry standard is essentially a concept that combines the advantages of non-contact mode imaging as well as contact mode imaging. That is, you use the cantilever as a spring, oscillate it, but do not oscillate it at a zone where it does not touch the surface. You let it oscillate, you bring it a bit closer, start oscillating and let it tap the surface like this. It goes on tapping the surface like this. So, what is the advantage? Advantage is, under this condition, let us say you have approached here, again I will discuss a bit on how to approach in a intermittent contact mode, and what happens is, suppose this was the free amplitude of oscillation, and when you bring it in close proximity to the surface, this already gets truncated. So, let us say this is 90% of the free amplitude. Now, you move on to your next point, let us say this is the point 2, you see that the surface is the point 2 is at a lower elevation as compared to point 1, right. So, what happens is this amplitude, let us say, now becomes over here 94% of the free amplitude, because there is simply more space. And now, what you do? You essentially choose a set point, let us say just the way we had a force set point in contact mode, you choose a set point, let us say, which is 90% of free amplitude, and now you know what to do, right. What to do? This is your set point, now your free amplitude has gone up to 94%. So, what you do? This information in the form of the error is fed to the piezo through the feedback loop. The piezo sort of will change its geometry, push down the holder or push down the location of the cantilever, it comes down, so that your amplitude is back again to the 90% or the set point amplitude. So, this is something I will discuss in a bit more detail. This is the intermittent contact or the tapping mode imaging. Geometry is roughly same, as a system is same, only excepting the fact that there is an additional drive at the to produce the oscillation of the tip. Most commercial AFMs come with the option that you can do a frequency sweep and find out what is the resonant frequency. So, what you do is you simply do a frequency sweep and you find out the resonant frequency of your tip you are using. Typically, the manufacturer specifies something, but every tip, because every tip has different dimension, different geometry due to manufacturing tolerances. So, it is a good practice to find out, and any reasonable instrument will allow you to do that, to find out the resonant frequency. Once you have found out the resonant frequency, so before that I must tell, up to the alignment is same. You would like to align, so I think I will draw it here again. Anyway, sorry, picture is becoming a bit bad. Anyway, after alignment, the laser spot falls at the center of the cantilever. So, after you have aligned, you essentially do your frequency sweep and choose. So, there is a drive motor, additional drive motor, which will oscillate your cantilever tip, and you choose certain fraction of the power, some power for the drive motor. So, what will this drive motor do? Oscillation, the amplitude is chosen by the resonant frequency. This drive motor will decide whether you would like to have higher amplitude or lower amplitude. So, for example, this, if your amplitude is high, it is obvious that you are going to choose a higher drive percentage. If the amplitude is low, it is obvious that you are going to choose lower percentage. So, now what happens? So, sample surface is somewhere over here, and sample surface is somewhere over here, and you have a cantilever that is oscillating. Okay. Now, as a consequence of this oscillation, what in fact happens at the QPD? That is an interesting question to ask, because unlike the contact mode where the cantilever was stationary, and therefore, at the beginning of the approach sequence, light was anyway falling at the center of the QPD. But here maybe I will draw a bit exaggerated view for easy understanding. After alignment, light was falling here, but the moment you turn on your oscillation, what starts to happen is the laser spot also sort of starts to oscillate over the QPD like this. So, there is a span, and let us say this span depending on the magnitude or the amplitude you have chosen. So, this range, what I mean to say is, if you have chosen a smaller amplitude, this is going to be the range. If you have chosen a larger amplitude, this is going to be the range, right, and it has to pass through the center. So, now the tip is oscillating and the approach sequence again with the help of the stepper motor begins. So, you are now bringing the sample and the tip close to each other. Again, it is the same, you can either take the sample up or can push it down, but when it is close to the sample, let us see what happens. What happens is, first it starts to tap, and then as you bring the sample closer and closer, let us say this is the configuration, it now, since it is tapping the sample surface, it fails to execute the whole entire oscillation during its amplitude, right. So, what happens now on the QPD, you see the oscillation is sort of truncated like this. This portion is truncated because of the presence of the surface, right. And now you simply choose your stopping criteria during approach as a percentage of the free amplitude. So, let us say you choose 90% of the free amplitude as the stopping criteria. So, when this area becomes 10% of the entire oscillation amplitude, the cantilever will the approach will stop. What you now all understand what is meant by approach will stop, the piezo in fact gets triggered, and then you can start scanning. So, now from point 1 to point 2, I have already discussed what is going to happen. So, you have an oscillating cantilever at point 1, which now due to the rastering goes to the second point. Let us say you have chosen set point to be %90, you move on to point 2, which is at a lower elevation. So, it is at a lower level. So, essentially the free amplitude, the amplitude, not the free amplitude, the truncated amplitude, which was 90% here, let us say it has gone up to 96%. So, that whatever is that equivalent, that difference between 96% and 90% on the QPD, essentially it translates to some amount of voltage. Suppose this was 90%, now you have gone up to 96%, so it has increased. Please remember, it has to pass through the center. I am just drawing them side by side. So, this is now the error, and this error is fed to the scanner, to the piezo scanner, so that that can take necessary actions. And again, once it is pushed back here, it is again back to the 90% or the set point value. So, one can ask you whether you stop at 85% versus 95% of the free amplitude, what would be the implication? Implication is, here the approach is a much softer approach, and here you are essentially tapping or knocking on the surface much harder. Okay. So, that is in a nutshell the tapping mode. I mean, you can see these pictures, and so this now should be clear to you. This is, let us say, the zone corresponding to the free amplitude. This gets truncated, it actually gets truncated from one of the sides. So, it is the reduced amplitude, and you have a set point, and based on that from point to point the amplitude is maintained to be constant. So, advantage, pretty high lateral resolution, no damage to the sample and to the cantilever tip, can image fairly rough samples, and ideal for sampling. This is wrong, in fact, ideal for sampling soft images. Resolution is a bit poor. So, it is almost time to wrap up AFM. Just one quick slide, I would like to remind you that AFM can be used for doing a whole lot of additional measurements, like let us say STM we have already talked about. One can do magnetic force microscopy, electrical force microscopy, atomic lateral force microscopy is something you have already told. We can also measure the interaction force between the tip and any location of the surface. This particular transparency will be available to you as a resource. There are some bullets on some of these advanced techniques, but I would not discuss them, because I do not want to make this course too heavy, and therefore, I stop my discussion on AFM right here. There are certain advantages of the AFM, but the salient points in simple words that I would like to highlight is, unlike other microscopes, AFM does not take a picture. It actually goes, there is a probe firstly that goes to the surface and does some measurement, and based on that measurement, in fact, you generate an artificially data rendered picture. So, in fact, the way you visualize in an AFM is completely different than any other conventional microscope. So, in very, very simple terms, while you see in other microscopes, you actually touch the sample, and therefore, you get a much better idea about the surface using an AFM. And since you are touching, just the way you can touch something and you can feel whether it is hard, soft, etc., etc., the AFM tip can also perform those functions for you and can give a lot of information. So, thank you very much. I hope you have some basic idea about the atomic force microscope now, and those of you who want to do further reading on this are most welcome. There is a huge amount of literature that is available in an AFM, and if you really take up nanotechnology as a serious career option in research, I am sure you will come across the AFM again and again and again, and these lectures, many of you will then remember, sort of gave you a first glimpse of the utility of an atomic force microscope. Thank you very much.